The key part of this is the image that you have been provided with the electron micrograph. So have a look at the types of things that you've got indicated in the diagram. Now it could be a case where you're not given these and you have to interpret the diagram for yourself. But in this case, the information is given for us. So this is just knowledge. Determine with a reason whether the image is prokaryotic or eukaryotic. So in this, because we have mitochondrion, because we have vesicles, because we have a nucleus, then we will indicate that our cell is a eukaryotic cell. It's a multicellular, it has endoplasmic reticulum. So that's what we're looking at to make the decision as to whether we have a prokaryote or a eukaryote. For the next question, state the process that divides one nucleus into two genetically identical nuclei. Mitosis. That's knowledge. That's covered. We need to think now that there are approximately 4 marks left for us in our next section, where we are explaining how the cell cycle is controlled. So in this, marking points could include, firstly, we have a sequence, so we go through the G1, the S, the G2, and mitosis. It's controlled by cyclins. We have an increase in cyclins and we have a decrease in cyclins. So what we actually have is fluctuation. We have surges, and these surges occur at different times within the cell cycle. So we've given an indication of the sequence. So we've discussed how it's controlled. We've looked at how the cyclins go up and go down, how they change throughout the cell cycle. The next point that we could discuss is that the inside and the outside conditions, both of them affect the regulation of the cell cycle, because we're looking at the control of the cell cycle. We're looking at regulating the cell cycle. Well, if we alter the outside or the inside conditions, we're going to have a change in that cell cycle. So this here is also worth a mention. We can go into more detail with relation to our cyclins and say, okay, well, we have 4 different cyclins, or we have different cyclins, and these are required at different stages of, or different events in the cell cycle. So here we're being a little bit more specific by saying 4 cyclins entering at different stages, and that they're necessary for the regulation of the sequence. So the sequence itself is determined by those 4 different cyclins. It has to be clear that the different cyclins are affecting or are acting at different phases. So I've put this in capital letters here. If we talk about the fact that we have them and that they are regulating, that might be enough for a mark, depending on how your sentence is structured. If you make it absolutely clear that the different cyclins are used for different stages or different phases, then you can be sure that you've been credited with that mark. Following on from that, we've talked about the regulation, we've talked about the order, we've talked about the cyclins and how they change. The next thing really would be to start discussing the fact that we have the cyclins that bind to kinases to activate them. So now we're not just looking at the cyclins, we're looking at how the cyclins themselves are activated. So following through a process. So the activation comes when we have the cyclins binding to the kinases, or the cyclin-dependent kinases. The kinases themselves phosphorylate the other proteins. So these kinases are phosphorylating the other proteins, and this allows for the cell cycle to progress. And the phosphorylated proteins perform very specific functions within the cell. So we've got the kinases. The kinases are needed to phosphorylate the proteins, and the proteins perform specific functions in the cell cycle. So these 3 things here, again, are quite intrinsically linked together. So we were breaking it down into the basics. We've got a sequence, we've got cyclins. Then we're looking at what happens to the cyclins in quite a low level. They fluctuate. And if we change the internal and external environment, we will have changes. We're then looking at it in a little bit more detail when we're saying we've got 4 specific cyclins, we've got regulation of different phases, We have got the cyclins binding to the kinases, the kinase phosphorylating the proteins, and the proteins performing specific functions. So these here, just as bullet points, very clearly outline what's happening in the cell cycle and how the cell cycle is controlled.